Welcome, everyone. Today we will be discussing Bacillus, a genus of rod-shaped, gram-positive bacteria. We'll look at the bacteria's morphology, cultural characteristics, biochemical characteristics, host range, virulence factors, and mechanisms of antibiotic resistance. Let's get started. Bacillus is a diverse genus of rod-shaped, gram-positive bacteria found in soil, water, and air. Endospore formation allows them to withstand extreme environmental conditions. Generally 1 to 5 micrometers in size, they also possess the ability to produce catalase enzyme which breaks down hydrogen peroxide. These bacteria are utilized in probiotic formulations to improve the health of humans and animals. In summary, Bacillus is an incredibly remarkable genus of bacteria. Bacillus is a genus of rod-shaped, gram-positive bacteria which can be found in soil, water, air, and gastrointestinal tracts of both animals and humans. It is remarkable that various species of Bacillus can survive in various conditions, such as highly alkaline and acidic habitats. Bacillus species are rod-shaped, typically ranging in size from 1 to 5 micrometers. Gram staining shows them to be gram-positive, where they retain the crystal violet stain. These bacteria also have the ability to form endospores, which are resistant structures that allow them to survive in harsh environments. Bacillus species have an extensive range of habitats, allowing them to form colonies on various media such as nutrient agar, blood agar, and McConkey agar. Colonies developed by these bacteria are generally smooth and opaque while emitting a particular odor. Additionally, they are obligate aerobes, requiring oxygen in order to proliferate. Bacillus species are known for their ability to perform a range of biochemical reactions. These include catalase production, glucose and lactose fermentation, starch hydrolysis, and gelatinase production. Catalase production is essential for breaking down hydrogen peroxide into water and oxygen. Within the group, most species can also ferment glucose and lactose, hydrolyze starch, and produce gelatinase, distinguishing them from other bacteria. Bacillus species have varying capabilities when it comes to infecting hosts, encompassing plants, animals, and humans. These numerous host varieties offer the potential to induce diverse infections. Additionally, certain Bacillus species are beneficial to humans and animals and are referred to as probiotics. The range of hosts that can be infected differs between various types of Bacillus bacteria. While some are able to target specific hosts and cause more severe infections, others have a more general host range and are often used as probiotics. Bacillus species can pose a significant health risk due to their ability to produce toxins, such as anthrax toxin and cereulide. These toxins can cause severe disease and can be particularly harmful when inhaled, ingested, or come into contact with the skin. Biofilms formation can also make them more resistant to antibiotics and immune defenses, making the potential effects more severe. Awareness of the virulence factors of Bacillus species is essential for protecting against them. In order to effectively manage the treatment of infections, it is important to understand how antibiotic resistance occurs within a bacteria. Resistance can develop through a range of mechanisms, such as mutations affecting target genes, decreased uptake of antibiotics, and increased pumps blocking the entrance of antibiotics. Epidemiology of Bacillus is an integral component when studying this genus of bacteria. As Bacillus species are found naturally in soil, water, and air, transmission can take place through contact with contaminated surfaces or inhalation of contaminated air. Additionally, some species, such as Bacillus anthracis, can be spread through contact with infected animals or animal products. Knowing the ways in which these bacteria can be spread is crucial to controlling its transmission. Bacillus species have been linked to a range of afflictions, from the life-threatening anthrax to minor foodborne illnesses, to skin and soft tissue infections to other serious maladies. Virulence factors are integral in determining the severity of the resultant disease, as the presence of certain genes can have a direct influence on the intensity of the infection. Regarding the immune system's response to bacillus infection, production of antibodies and activation of T cells are both activated. Both the innate and adaptive immune systems collaborate to create an effective response to the pathogen. Culturing bacteria from blood or tissue samples and serological tests such as PCR-based assays are the primary laboratory tests used to determine if a patient is suffering from a bacillus infection. Imaging studies may also be used in conjunction to make a definitive diagnosis. Proper food handling, preparation and hand washing are essential in preventing infection caused by bacillus. Vaccines are also available to protect against some species, like Bacillus anthracis. Implementing these measures helps minimize the risk of infection and increase the chances of staying healthy. Treatment of infections caused by Bacillus bacteria must be targeted to the species of Bacillus involved. Antibiotics are the primary treatment option and can include penicillin or tetracycline. In addition, supportive care such as oxygen therapy and intravenous fluids may be necessary. Bacillus is a diverse genus of gram-positive bacteria with a wide range of habitats and virulence factors. These bacteria can cause severe diseases in humans and animals, or may provide health benefits as probiotics. Prevention, diagnosis, and treatment of bacillus infections are essential to protect public health. Thank you for your attention.